All right, so I'm going to talk today about skeletal muscle contraction. And in order to start skeletal muscle contraction, the skeletal muscle fiber itself has to be excited by the nervous system. So before we even start, we have to talk about neural control. Okay, so when we talk about cells, we have a plasma membrane, right? That's separating the inside of the cell from the outside of the cell. So the inside has a negative charge to it, and the outside has a positive charge. Now, this is really the separation of charges. This cell is polarized, okay? And this potential difference across the plasma membrane is the resting membrane potential. Okay, so the negative charge on the inside is due to ions that are on the inside, but mostly due to big proteins that really can't move across the membrane. So the resting membrane potential in a neuron is about negative 70 millivolts, okay? Now, we keep this resting membrane potential. This is due to pumps, specifically the sodium potassium pump. Okay, so it's pumping sodium and potassium across the cell, and what it's doing is it's giving us a concentration gradient. Now, the reason it does that is so we can build up certain things on the inside and certain things on the outside. So actually what we're building up on the outside is sodium, and what we're building on the inside is potassium. So we have a lot of sodium on the outside of the cell and a lot of potassium on the inside of the cell, okay? So that sets us up to create some kind of excitement for the skeletal muscle cell. So what happens? Draw our famous little chart here, okay? So we're at negative 70 here. This is resting membrane potential, all right? So say something happens, okay? Something happens, the spider's on your leg, something to where you're gonna need to move the skeletal muscle, okay? So you go from negative 70 and you create a potential, so a change in what's happening here, and you get to a negative 55 threshold. Well, action potentials are all or nothing. So once you get there, you basically, what happens is you open sodium channels, okay? So if you open a sodium channel has a positive charge, and sodium's going to rush into the cell, okay? Now, when a positive charge comes into the cell, which is negative, you go from being very negative to being a positive number, right? Because you're adding positivity into the cell. So now you've gotten up to 30, okay? Positive 30 millivolts about. These are millivolts, okay? So now what you do is you close the sodium gate and you open the potassium channel. Now, if you remember where potassium was stockpiled on the inside of the cell, okay? Now what we do is we let potassium rush out of the cell, okay? If you let a positive charge leave the cell becomes more negative, right? You're giving away all your positivity, so you become more negative again, okay? So this event over here is depolarization. This over here is repolarization. And both of these together equal an action potential, okay? The action potential is like a lightning bolt, or like the energy that's going to ignite the skeletal muscle fiber. So what you did, so just to review, you stockpiled sodium on one side and potassium on the other, and you did that via the sodium potassium pump to give you a concentration gradient, okay? The reason you created a concentration gradient was so that you had a buildup, so you were able to open a sodium channel. If you hadn't stockpiled here and you tried to open a sodium gated channel, nothing would flow, right? Because you wouldn't have anything there. So you use this pump to create a concentration gradient so that when you had to change from the resting membrane potential of -70, right, you could open a voltage gated channel, allow sodium to fly through, right, and change to a positive number. So this whole event, depolarization, repolarization event, is an action potential. Okay, so where does the action potential go? Well, the action potential is traveling to the skeletal muscle fiber, okay? Now, where does the skeletal muscle fiber and the neuron meet? So skeletal muscle fiber plus the neuron, they meet at the neuromuscular junction, okay? So they meet at the neuromuscular junction. So what you're gonna have is we're gonna have our neuron here, okay? And then let's do our skeletal muscle fiber in a different color. We're gonna have our skeletal muscle fiber down here, okay? So skeletal muscle fiber. And in between is this space that we call the synaptic cleft. So this is the space in between the neuron and the skeletal muscle fiber. So it's where they meet, okay? So the action potential that you created over here travels, okay? And it travels down the neuron, okay? Now what does it do here? Well, in the neuron you have little vesicles, okay? And the little vesicles hold acetylcholine. Acetylcholine is a neurotransmitter. A neurotransmitter is anything that's going to transmit a signal, okay? So ours here is acetylcholine. Now, for acetylcholine to have any activity, it has to have a receptor somewhere else. So it makes sense if we're trying to activate a skeletal muscle fiber, the receptor for acetylcholine sits on the skeletal muscle fiber, okay? So what happens when the action potential travels down to the neuromuscular junction, it causes the release of acetylcholine. And when acetylcholine is released, it comes and binds to its receptor. Now what happens when it binds to its receptor is you open a sodium channel again. Now this time the sodium channel is no longer voltage gated, it's ligand gated, but nevertheless you've opened a sodium channel. And you remember what happens when you open a sodium channel, right? You get your depolarization, repolarization, you get the action potential, okay? So you get that lightning bolt, okay? So now you've successfully turned an electrical signal created in a neuron and you've put it in a skeletal muscle fiber, okay? So now you have excited the skeletal muscle fiber, okay? So you've propagated the action potential down into the skeletal muscle fiber, okay? So now where is it going, okay? So we're in the skeletal muscle fiber, the action potential travels into the fiber, and where it's actually going to travel into is the sarcoplasmic reticulum, okay? So we have our skeletal muscle fiber here, okay? And you have these little things that come down. They're called T-tubules, okay? And T-tubules travel down. I'll just kind of draw what looks like kind of a web here. T-tubule. And this is the sarcoplasmic reticulum, okay? Now the very special thing about sending the action potential here is that the sarcoplasmic reticulum stores calcium. It stores it in something called terminal cisternae, okay? But just know that calcium storage is in the sarcoplasmic reticulum. So when you send the action potential down into the skeletal muscle fiber, into the sarcoplasmic reticulum, you release calcium, okay? So you spill calcium out everywhere. It's like spilled milk, okay? So calcium spilled absolutely everywhere, okay? Now, the very, very special thing about calcium is it has a specific attraction to the myofilaments that lie within the skeletal muscle fiber. So let's draw some of the skeletal muscle fiber components. So we'll just kind of zoom up here. Let's find our red. So if we were to look underneath in here, you'd see all your myofilaments in here. So I'll just kind of draw them. They lie underneath. So if we were zooming underneath the sarcoplasmic reticulum, we'd see them. And they are actin and myosin. Okay? Actin is the thin strand, myosin is the thick strand. So actin and myosin, when they bind together, they do something called the sliding filament theory, and that's for muscle contraction. Now the thing is, they can't bind together really. Actin looks kind of like a twisted strand of pearls, and it has things blocking it. It has guard dogs, okay? So it has a little rope on it called tropomyosin, and it also has something holding its rope in place called troponin, okay? Now, there's no way myosin can bind to actin with all these guard dogs in place. So the really special thing about calcium is calcium has a specific attraction for troponin. So when this action potential comes down into the sarcoplasmic reticulum and releases calcium, calcium goes and binds to the troponin. So when calcium binds to the troponin, it moves not only troponin, it moves the tropomyosin out of the way. So now all the active sites on actin are open and ready for binding. So at this point, let's just draw our little myosin. This is one myosin with a little cleft head. So at this point, as long as you have, you remember it, as long as you have ATP, right? This myosin can bind to this actin, okay? And they'll do something called the power stroke, and I'll do that in a different video, but that's basically the basics of skeletal muscle contraction. So let's talk about it one more time. So we started in a neuron, right? A neuron has to excite a skeletal muscle fiber. So how we created this concentration gradient was we used the sodium-potassium pump. We stockpiled on either side, okay? And we had a resting membrane potential of -70 in a neuron. So that's when nothing's happening, okay? But say you have some sort of stimulus, okay? What you do is you open a voltage-gated channel. So the voltage-gated sodium channel allows sodium to take its positive charge and fly into the cell. That adds a positive charge to a cell that was negative, okay? So you start to move up on this scale of millivolts, right? And that's called depolarization. Okay, well, once you reach a threshold, about +30, open those potassium gates, okay? So what you're gonna allow is you're gonna allow all the potassium with its positive charge to leave the cell. If you take a positive charge away, you become more negative again. So that's called repolarization. Both of these together create an action potential. That action potential is like the battery voltage or the electricity, and it travels down the neuron to meet a skeletal muscle fiber, okay? When it travels down, it releases a neurotransmitter. The neurotransmitter binds, it crosses the synaptic cleft, and it binds to a receptor on the skeletal muscle fiber. This helps you propagate the action potential now in a skeletal muscle fiber. So we're no longer in a neuron anymore. We have propagated the signal down to the skeletal muscle fiber. Where is that going? It's going into the sarcoplasmic reticulum via a T-tubule. So it travels down the T-tubule into the sarcoplasmic reticulum, and it causes calcium to be released. The big deal about calcium is calcium has a specific attraction or affinity to tropomyosin, or to troponin. Troponin and tropomyosin are guard dogs that sit on actin, okay? Actin and myosin are your myofilaments. They are what do the muscle contraction. But the thing is actin is blocked. Its active sites are blocked by these guard dogs. But when you release calcium, calcium binds, moves them away, and as long as you have ATP present, myosin and actin can bind and do their power stroke, and muscle contraction can happen. So that's your basics of skeletal muscle contraction.